Welcome everyone. Today in this class we'll deal with this last plant growth regulator, that is abscisic acid. We have already seen auxin, gibberellin, cytokinin and ethylene. Out of that four, auxin, gibberellin and cytokinin is a they are growth promoting substances, whereas ethylene and this abscisic acid, these two are growth inhibitors. Earlier it was said that this abscisic acid is only responsible for abscission and seed dormancy. It is responsible for abscission and seed dormancy. Abscission is nothing but removal of that or dropping of the plant organ, that is plant part, dropping of the plant part from the plant, and seed dormancy, that is the condition where the seed will not be able to germinate, the condition where seed will not be germinating. That condition is called as seed dormancy. Abscission, removal of a particular plant part, whether it can be leaf, fruit or flower from the plants. So this work, that is abscission and seed dormancy, was done by abscisic acid, which is also called as ABA. The short form of it is ABA. ABA is nothing but abscisic acid. Earlier it was said that it was discovered that it is only helping in abscission and seed dormancy. Later it was found that as the other organ, that is growth hormones, they are also performing other different functions. In the similar manner, this abscisic acid can also perform various other physiological functions. Apart from these two, it can also perform other physiological functions. Here it acts as plant growth inhibitor. It will inhibit the growth, it will stop the growth of the plant. The plant cannot continuously grow. At some stage it needs to stop the growth. The plant growth needs to be stopped. So that stopping, for a while the plant growth need to be stopped. The old leaves need to be shed. The old flowers need to be shed. The old fruits need to be shed. Here, so that process is the inhibiting of growth, inhibiting of plant growth, that is the shedding of leaves, the shedding of flowers, the shedding of fruits, that is that is changing the leaves before falling, it will change to pale color, then it will drop off. So all these are the plant growth inhibiting activities. All these are the plant growth inhibiting activities, and that inhibiting activities is done by abscisic acid. So abscisic acid is acting as plant growth inhibitor. It not only inhibits, stops or prevents the growth of the plant, but it also inhibits the plant metabolism also. Whatever the reactions which are carried out in this inside the plants, that will also be inhibited by abscisic acid, because naturally when this plant metabolism is inhibited, whatever the reactions and all which is being carried out inside the plants, when it is inhibited, stopped, then the growth of the plant will also be stopped. So apart from this breaking dormant, that is apart from seed dormancy and abscission, it also inhibits the metabolism of plant as well as the growth of plant. Here it also inhibits seed germination. It will not allow the seed to germinate. It will not allow the seed to germinate. So that means it causes seed dormancy. Seed dormancy is nothing but the seed will be unable to germinate. The seeds will germinate and produce plants. So here when the seeds cannot be germinated, that condition is seed dormancy. When seed dormancy is there, then seed germination will be inhibited. So that is why So this abscisic acid is acting as the inhibitor of seed germination. Then it stimulates closure of stomata. Stomata, we all know what is stomata. Stomata is nothing but the small holes which is present on the leaves through which the water will be coming out. The carbon dioxide will be going inside, the oxygen will be coming outside. So these are the small holes which is for gaseous exchange. The holes which are present on the leaves, stomata are nothing but pores on leaves. They help in gaseous exchange and transpiration. Gaseous exchange and transpiration. So here it stimulates the closure of stomata. It allows the stomata to close. The stomata, when it opens now, then only gaseous exchange will take place and then only transpiration will takes place. But for some reason now, the stomata needs to be closed for a period of time, for a certain period of time. Plants continuously needs oxygen, that is oxygen will be coming inside. If plants are not performing photosynthesis, then oxygen will be coming inside and carbon dioxide will be going outside. But if the plants are performing photosynthesis, then carbon dioxide will be coming inside and oxygen will be going inside. There will be continuous exchange of gases based on which process they are performing. So if the exchange of gases is taking place continuously, then stomata needs to be open. So but for certain reasons, the stomata needs to be closed. So that closing of stomata, that stimulation of closing of stomata is done by abscisic acid. So why the stomata needs to be closed? Why stomata needs to be closed? What happens is, this stomata will be closed in order to provide tolerance to the plants, tolerance of stress to the plants. In order to tolerate the stress by the plants, here the stomata will be closed. For example, if the temperature is very high, so with very high temperature, the water transpiration will be very high. So if water is transpirated, that is, if transpiration is taking place at a very high rate, then the plants will be they will be facing deficiency of water, especially in drought conditions. Where this water is, there is scarcity of water. Already there is scarcity of water, and whatever water is there in the plants, with this high temperature, if the stomata is open, the plant will be transpirated, that is, the water will be transpirated. So in order to avoid the transpiration, because if transpiration takes place, then the plants will lose the water. So in order to avoid this losing of water, the transpiration needs to be stopped. In order to stop the transpiration, the stomata needs to be closed. Because if stomata is open, the water will be transpirated. So in order to stop the transpiration, because of the various conditions, that is tolerance in the sense, in order to tolerate the drought, then the stomata needs to be closed. So with the help of this closing of stomata, the plant is able to tolerate that stress of the drought. That is why this hormone, this plant growth regulator, abscisic acid, is also called as stress hormone. It is also called as stress hormone. So it is because it is helping in tolerating the stress which is caused due to drought by closing of stomata, by the stimulation of closing of stomata. So that is why this plant growth regulator is also called as stress hormone. Now apart from this, it also helps in seed development. When the seed is in dormant condition, that is, it will be causing seed dormancy. But when the seed is when the seed has came out of dormancy, when it started to germinate, then the development of that seed after it germinates, when there is no dormancy, after it germinates after germination, then the development of seed will have to take place. So in that development of the seed, abscisic acid will be helping. Abscisic acid will be helping in seed development. Seed development takes place when the seed comes out from dormancy. After seed comes out from dormancy, because of various reasons, the seeds will be coming out from dormancy and it will germinate. So when it germinates it needs to be developed from that seed, it needs to be developed into a whole new plant. So the development of the seed is done by abscisic acid. The development of seed, maturation of seed is done by abscisic acid. Okay, and if the conditions are unfavorable, then it will cause seed dormancy. If the conditions are unfavorable, it will cause seed dormancy. Apart from this, it also acts as antagonist to gibberellins. It is showing opposite effect to gibberellin. Whatever gibberellin is, if it is promoting, then this abscisic acid will act as the antagonist. It will affect in opposite way. So to summarize about all these growth regulating hormones, so here at every stage of growth and differentiation, that is during the development of plant, there we see growth and differentiation, and every phase of this growth and differentiation, one or the other hormone is helping. One or the other hormone is playing its role. For example, to initiate cell division, to initiate the growth, we require auxins and cytokinins. And to initiate seed dormancy, we require abscisic acid. Then to initiate ripening of fruits, we require ethylene. To elongate the plant parts or the fruits, we require gibberellin. So here these plant growth substances at the starting, if the seed is unable to germinate at the starting and during seed dormancy, abscisic acid will help. Then during after the seed is germinated, the roots and shoots will be coming out, auxins and cytokinins will be helping. Then to increase the length of the plant, gibberellins will be helping along with auxins and cytokinins. Then after it is grown, the fruits, flowers and all will be produced. There also auxins and cytokinins will help. Then after formation of fruit, then the fruit will be ripening. Then there ethylene is required. Then after ripening of fruit, the fruit needs to be dropped off. Then abscisic acid is required. So at every stage of plant, every stage of growth and differentiation, one or the other hormone has a role to play. So these hormones can be both the two hormones can be complementary or antagonistic, opposite. For example, the gibberellin and abscisic acid, they work in contrast, they work in opposite. If it is helping, if gibberellin is promoting, then this will inhibit the growth. And they can be complementary in the sense they can work side by side. For example, the growth, for the promotion of growth, auxin and cytokinin, they will be helping side by side. Both of them will be doing the work simultaneously, helping one another. Auxin will be helping in cell division, cytokinin will be helping in, so that is promoting growth by producing number of cells. So yeah, they can be complementary to each other. Example is auxins and cytokinins, and they can be antagonistic, that is they will be working in opposite, and they can be working individualistic. Only single hormone will be working at a particular time, or two hormones together will work. So there can be individualistic, that is only one hormone will be working at one time, or else sometimes both hormones, two hormones will be combining and working. So but growth regulators have a massive effect on the growth and, that is growth and differentiation of the plant. The PGRs has a massive role in the development of the plant. Apart from this, here this PGR, plant growth regulators, are only one intrinsic factor. They are only one intrinsic factor. Apart from plant growth regulators, the genomic factor is also there. The growth of the plant, the differentiation, that is the development of the plant, is dependent on two intrinsic factors. Here factors are of two types, intrinsic and extrinsic. Intrinsic and extrinsic. Here in intrinsic now two factors are there. First is PGRs and second is genome. So the growth is dependent on the DNA of the plant, that is genome of the plant, as well as on the plant growth regulators of the plant. So these two are the factors which are present inside the seed itself. And other extrinsic factors which helps those are temperature, light, gravity, all these are the other factors, that is those are the extrinsic factors. So in intrinsic factors, the one was the important one. Apart, that is both of them are important, but here this plant growth regulators have a very important role to play in the development of the plants. Next we will deal with seed dormancy. With this we have finished all the 5 growth regulators. Next we will go with seed dormancy. In the next class we will see seed dormancy.